Hello, my name is Xiang Fu, Professor of Cellular and Molecular Medicine at the University of California, San Diego. When Berlin Wall fell in November of 1989, I was actually in the middle of making the discovery of the first protein factor called SC35, responsible for committing pre-mRNA to the splicing pathway. This marked the beginning of my lifelong journey in understanding RNA biology, which has led to today's discovery in converting non-neuronal cells into neurons by repressing a single RNA binding protein called PDV, now published this year in Nature. Let me make a few slides to make my point during this presentation. The bottom line here is to make new neurons in the brain to replace lost ones in the disease settings. Doing our basic research for regulated RNA processing, we accidentally discovered that reducing the expression of PTB convert literally all cell types we tested to neuronal-like cells, as exemplified here on astrocyte, a major supporting cell type to neurons in our brain. Interestingly, astrocytes become proliferative when neurons suffer from damage. The idea here is to convert a fraction of those extra astrocytes back to neurons. This finding now opens new doors to making new neurons to replace lost ones in brain as a strategy to treat your degenerative disease, a problem that faces the entire humanity, especially in aging societies. This problem is particularly challenging as the existing neurons themselves cannot make new neurons, and the neurons become dysfunctional way before death. This is why many existing approaches are not effective when disease symptoms already start to appear. We now applied our new strategy to a mouse model of Parkinson's disease, which is due to the loss of dopaminergic neurons, and found a single injection of our reprogramming agent is sufficient to rebuild the lost dopamine circuitries. As we can appreciate from the images, we selectively killed dopaminergic neurons labeled in green in the right side of the mouse brain, shown in the middle, leaving the entire left side as a control. After reprogramming, the lost neuron were successfully restored, as illustrated on the right. The mouse become normal after induced neuronal reprogramming, as shown in the rotation assay. Here, because one side of the brain was impaired, mouse rotate in one direction, clockwise in this case. And after reprogramming for 3 months, this motor neuron phenotype completely disappeared. Because astrocyte could be converted to different subtype of neurons during our basic research, which indicate our strategy may be generally applicable to treating a variety of neurological disorders. We obviously have a long journey before clinical trials by testing on multiple different genetic models for Parkinson's disease as well as other diseases. We need to develop a strategy to inactivate existing disease mechanisms, overcome age restriction to neuronal reprogramming, and determine potential side effect. Most importantly, we need to test on our strategy on non-human primate models before clinical trials. It would be a dream coming true if I'm able to see real benefit on patients soon. After publishing our findings in Nature in June this year, I've received numerous phone call from patients, and now sense the real urgency more than ever to push our discovery toward such a goal. Thanks for listening to the story. Again, it's such an honor to be nominated for the Falling Walls Breakthrough of the Year in Life Science. Thanks.